Hi, my name is Maria Alejandra Ariza. I am from Colombia. I'm a biologist from Pontificia Universidad Javeriana, and my work is entitled In Silico Comparison of the Effect of Extracts of Passiflora edulis and Lippia alba on dendritic complexity in neuronal cultures. Well, my work is framed around the development and functioning of the central nervous system known as CNS, specifically to promote good cognitive function. Neuroplasticity, for this reason, is an important process for cognitive maintenance and is defined as the ability of the CNS to change its activity in response to intrinsic and extrinsic stimuli by reorganizing its structure, function, and connection. It should be noted that these neuroplastic regions are sensitive to several dysregulation, leading to a progressive loss of structure and function. This process is called neurodegeneration. However, some studies, like Spencer in 2009, have shown that some flavonoids present in plant extracts can have a neuroprotective effect. For this reason, we consider important for this work to study the secondary metabolist of Passiflora edulis, known as gulupa, and Lippia alba, known as pronto alivio, and study if their secondary metabolist or flavonoids contain this neuroprotective effect. Also, we consider important to study the whole complexity of the dendritic tree of the neurons. For this reason, we made a code based on DLA, that is a diffusion limited aggregation, and an index called SDI, that is a shape diffusiveness index, proposed initially by Lusac in 2010 to actually compare the effect of both Colombian plant extracts and measure their complexity. For this reason, or for make all of this, we made a code and we run the code in 35 images per treatment. We had 3 treatments, the Passiflora edulis, the Lippia alba, and routine as a positive control. Each each treatment we made with neuronal cultures of 18-day-old Wistar rat embryos, treated with, the treatments that I already said, and then we immunostaining the neurons against MAP2 protein for actually see the dendrites. Then we do the DLA algorithm implementation, in which we just put a seed in the center of the image and we basically reproduce the image that we already had by random walk. And then we use the SDI, that is defined as exponential minus d, to actually, understand the complexity of the figure. So values closest to 0 were for less complex structures, and values near to 1 were for more complex structures. So as a result, we have 2 important things to mention. First of all, the verification code. So we run this code into images, a line and a Purkinje cell, and we find that the code was working because we had a higher SDE value in the most complex figure. And in this figure 3, we can just see the box plot and we can see the distribution of the SDI and the different concentrations. And we just see that Passiflora edulis, that is shown in green, has the highest values of SDI. In conclusion, the code is effective, allows to compare the images and compare treatments, and to determine which is the more complex neuron. And of course, the highest complexity is obtained for routine, followed by P. edulis and Lippia alba. Thank you very much for watching this video.